Please excuse any issues related to the time. We have an interesting session today, and we're going to start off with Dr. Cristianne Rijcken, who's the founder and CSO of Cristal Therapeutics, and she will be talking about the clinical phase 1 and 2 evaluations of their drug CPC 634. Cristianne?
Thank you, Neil, for the kind introduction, and welcome from the Netherlands. So it's really a true global event, despite being virtual. But indeed, it's my pleasure to present Cristal Therapeutics to you, and more specifically to talk about the application of the Rijcken platform in oncology. So what is... That's a little bit too quick. So the key thing is still in oncology is that a lot of products still doesn't have optimal therapeutic outcome because of either too much toxicity or insufficient drug levels at the tumor site. And we, with Rijcken, aim to overcome those challenges by bringing more drug to the tumor, so more specific tumor targeting, and lowering the safety hurdles, so particularly improving the safety profile, so really shifting that balance towards a more optimal outcome for the patient. And aiming to develop different type of products for optimal tumor treatment. So the way how we do that is with Rijcken, and Rijcken stands for Cristal Therapeutics Polymer Technologies. So it actually already abbreviates polymer technologies, means in our case we use polymer to entrap drug molecules. More specifically, it's a unique approach, namely that we derivative the drug first with a linker molecule. So that's on the left side, you see the API linker molecule being generated. We combine it then with a polymer, and then we create this nanoparticulate structure in which the drug is inside the core of these nanoparticles. But even more importantly, as you may see, both the drug molecules as well as the polymer chain, they're all covalently linked to each other, meaning this is an intact nanoparticle. So initially really a 3D matrix molecule. But clearly over time, the native drug is being released as a result of chemical hydrolysis of the drug linker bond. So this is what we call the passively targeted nanoparticle, and if preferred, we can conjugate to the surface molecules, either for imaging, for targeting applications, and other applications. So what we've developed so far is these type of nanoparticles where we entrap the API linker molecules, and we actually nicely confirmed that the API is in the core of the nanoparticle by performing SACMAL's UV analysis, where we had a very nice overlap of both the API signal and the nanoparticle, really illustrating that the drug is nicely in the core of the nanoparticle. Secondly, if we then inject these type of nanoparticles into the bloodstream and compare it with the native API, what you then can nicely see is that the native API has a rapid declining blood levels, as is known for a lot of these molecules. And this is just showing you a representative model. So we have these data for quite some different type of APIs, small molecules. But if you then entrap the drug into nanoparticles, you really have a much longer blood circulation profile. So really preventing the rapid clearance and assuring long circulation. So the reason why we see this prolonged circulation is not only because we have a good entrapment of the drug in the nanoparticle and the temporarily covalent linkage, but we also, in collaboration with the Mainz University, demonstrated there was hardly any protein absorption to these type of nanoparticles. More specifically, was this done by a technique called AF4, so asymmetric fuel flow fractionation, in which particles that were incubated with blood plasma proteins are in a very mild way fractionated, meaning separated from the non-adsorbed proteins. And then thereafter, they were characterized by all kinds of different methods, including also DLS, particle size analysis, and then even at a later stage, also the extent of proteins were both quantified as well as identified. And in our case, what we were found, or actually what our collaborators in Mainz found, was there was hardly any protein absorption, also as illustrated by the minimal increase, or actually the absence of the increase of particle size. So you see 31 nanometer versus 30 nanometer. And even more importantly, if we quantified and identified the type of proteins, it was predominantly a very small amount of albumin that conjugated or adsorbed to this type of nanoparticle. And this is again also confirmed for all kinds of different nanoparticles. Then clearly the question is, where do the nanoparticle actually go? So what we've done in collaboration with RWTH Aachen University, we administered these type of nanoparticles to mice. More specifically, they were labeled with size 7, so a fluorescent dye. And then we monitored their biodistribution profile, as nicely could be visualized by these FMT CT scans. And we monitored that up to 20% injected dose of the particles did reach the tumor site, so per gram tumor. Even more importantly, if you would quantify the uptake in a non-invasive manner that was feasible due to the size 7, then you could nicely see that really predominantly in tumor tissue we saw uptake, whilst the other organs, they showed some initial uptake, but then it was lowered again, and that is simply because of the long circulation, so meaning the blood contribution to the initial signals. But particularly the liver and the spleen, I would say the known organs, they do clearly play a role in the accumulation, namely that they actually result in removal of these nanoparticles. But as said, true uptake is really in the tumor tissue. So then what we did as the next step is that take out these tumor tissues and take a closer look where the nanoparticle actually resides. And what we found is that the predominant part of, let's say, 2/3 of the nanoparticle still reside extracellularly after 2 days in the tumor tissue. So that means that for drugs that are in the tumor microenvironment, if the drug can cross the cell membrane, then you're already there. But what we also evaluated that, let's say, roughly 33%, so 1/3 of the nanoparticles that are really intracellularly are in macrophages, meaning that not only we can reach the tumor cells, but we can also get involved with these type of immune cells. So in a way, we show that the cryptic particles have a more preferential uptake in tumor tissue in these preclinical models, and do not only reach the tumor cells, but also the macrophages and other immune cells. So then, as I showed in the former slide, we conjugated compounds to the surface, and we've done that by a proprietary technology called Clicker, which is actually a copper-free click reagent. In the first case, you can use it to conjugate a label to it, but since we've shown that we have quite limited uptake in the in specific cells, we also envision that we can use it to go for active targeting. And by active targeting, I mean conjugating all kind of peptides or protein fragments to the nanoparticles. And next, as I said, we can also use this conjugation chemistry for imaging agents, as I will address it later on, but you could also envision to use it for vaccination, a topic that I will not discuss today, but which we at Cristal are working on these days. So via this active targeting approach, in this case, we conjugate a Fab fragment to the nanoparticles and then evaluate the selective interaction with either cells not expressing the receptor correlated to this fragment, or in case the receptor was present, and we saw nicely increased uptake in the target cells, all a result of this active targeting. So meaning all in all, if you can go for passive targeting, that can be a route, or go for active targeting in case you even want to have further specific cellular interaction. But then clearly, this is kind of the overall platform opportunities. Clearly, the key focus of the talk today is about the clinical development. And pleased to talk about the CPC634, as we call a clinical candidate, which is the nanoparticle comprising docetaxel, and more specifically, docetaxel nanoparticles that are roughly 65 nanometer in diameter. And to be very explicitly, these are non-targeted nanoparticles, so there is no targeting ligand on the surface. So what we've done is this phase 1 trial, which is a sponsor-driven trial, and a more traditional dose escalation approach, so 3+3 design. And all in all, we treated up to 33 patients, in which we penultimately could demonstrate very nicely dose linear PK profile, and even more importantly, a half-life of the nanoparticles of roughly 33 hours, 31 hours for the first time point, 60 milligram per square meter. And that is clearly much longer than the Tageted, but as you can see, also particularly for the AUC, it's really much higher, really representative of the long circulation of the nanoparticles. Well, clearly, a phase 1 is never intended to focus on efficacy readouts, but clearly we monitor them along, and we saw in these 33 patients quite some efficacy signals, stable disease, and also some partial responses. And if we move forward as a conclusion from this phase 1 trial, which was finalized already a year and a half ago, is that we, as compared to conventional docetaxel, which is branded under the name Tageted, we really had a significantly changed PK profile, like I showed you in the former slide, but even more importantly, the safety profile was really significantly improved. And particularly the focus was that we hardly saw any neutropenia, probably as a result of the really changed PK profile. So meaning that we didn't have the high Cmax, meaning the really exposure of the body to high docetaxel levels in the beginning. Namely, in the beginning, it was entrapped by the nanoparticle. And then what we define as the so-called recommended phase 2 dosing, we define that as being 60 milligram per square meter every 3 weeks with dexamethasone pre-medication. All because of what we saw long-term safety profile based predominantly on the skin tolerability. So this was the profile with which we went forward to the phase 2 trial. Unfortunately, due to Corona, there was a slight delay in the results of the phase 2 trial. So unfortunately, I cannot disclose any results so far, since the data is still being progressed. Besides, recruitment has stopped, but now we're processing all the data. So that's still ongoing. Nevertheless, based on the, let's say, the concept of improved tumor uptake, we decided to start 2 investigator-driven trials. The first one being a trial together with Ron Matthijs at the Erasmus Hospital University. And that is a study that we called Crytex, since it is literally a crossover design study. And I think it's a unique study worldwide, namely where patients were given either first our product, so CPC634, or Tageted, or the other way around. And then at different time points, tumor biopsies were taken. So that means that every patient got twice a tumor biopsies after either first our product or Tageted or the other way around. So really each patient was its own control. And this really allowed for a very elegant evaluation of the actual tumor uptake in the patient, in a specific patient, in a specific tumor lesion. So then the results that came out of this trial were quite promising, but also partially surprising. Promising because of, as you may see here, the total docetaxel levels. They were roughly 450 times higher total docetaxel levels as compared to Tageted. So really indicative that the nanoparticles reach in a clinical setting 4-fold higher levels as compared to Tageted. But I would say the most surprising part was the fact that Tageted levels remained fairly high over up to 2 weeks of period of time. So we literally measured up to 2 weeks after one single injection. And for me, it was at least quite surprised that Tageted levels would really be persistently high along those 2 weeks period. I would have envisioned you have rapidly declining blood levels, so meaning you have limited exposure to tumor tissue, and due to the fragility of the docetaxel molecule, I would have expected degradation followed by elimination. But surprisingly, with a validated bioanalytical method, we've shown up to 2 weeks quite consistent high levels of the Tageted. So all in all, this demonstrated that we do have good uptake, but the differentiation versus Tageted is not demonstrated in this clinical trial. Clearly, taking biopsies is not the most preferred route going forward. So in parallel, we did another clinical trial and another investigator-driven trial, in this case together with Willemien Menke from the VUMC in Amsterdam, in a project that was sponsored by Brussels. And more specifically, in this case, we labeled, a PET labeled to the surface of the nanoparticles, enabling for non-invasive imaging. And this is what we've also done with this couple-free click chemistry. So then the study setup was as follows, namely that we first screened in patients the actual tumor lesions, the residence of the tumor lesions by FDG PET. Then thereafter, patients were treated with what we call tracer only, meaning a small amount of this radiolabeled nanoparticle, and we followed that up with a couple of scans. And only after that, we gave a therapeutic dose of the CPC634, so 60 milligram. And then within 1 hour, we also administered with a small amount of this radiolabeled version, and then followed up scans again over time. Then what we demonstrated in scans of these patients over time is that actually we saw also again in these patients a really prolonged circulation. So up to 20% of the injected dose was still present after 4 days. So really indicative of the long circulation like what we formerly have seen. And then the various organs, we saw a profile that was, I would say, resembling the preclinical data. Namely, we did saw some uptake in the liver as well as the spleen, the let's say, known organs of eliminations of these type of nanoparticles. And then clearly the key question is what happens to the tumor uptake? That was visualized with, for example, these representative pictures, where we show two patients, one with a tumor in the cheek, that in FDG there was clearly a tumor lesion present, and then we saw also nice elimination of the PET, so really representative that nanoparticles did accumulate in the tumor lesion. What we kind of quantified is that we saw in roughly 35% of the evaluable tumors we did see uptake. But I should make one remark here, namely that particularly in the abdominal region, so meaning in the region where a lot of lesions were, we could unfortunately not quantify the uptake. And why is that? Because in the former slide, you saw that there was quite some uptake of the nanoparticles in the spleen and the lesion, meaning in other words, the signal to background, the noise to signal value was a little bit distorted, meaning in the abdominal region, you couldn't quantify uptake in the tumor lesions. However, more importantly, what is the fact is in if you evaluate the microdose versus the therapeutic dose, that you could nicely see equal uptake levels in the tumor lesions. Meaning, if you interpret these data, that means that if you go forward with a small amount, so what we call the microdose amount, 1 milligram, that gives equal biodistribution profile alike with the therapeutic dose. So all in all, in the future, we envision that you could use a small amount, which is non-toxic because it's really a limited amount, and it really has a representative profile of the overall biodistribution of a therapeutic level. So patients do not need to be exposed to these therapeutic levels in case there is not sufficient tumor uptake.
Cristianne, 1 minute.
So yes, thanks. The summary is that by now we demonstrate quite good uptake into the tumor uptake, but clearly what we further need to work on is to assess optimal exposure in the tumor lesion, meaning really focusing on linker part. For that, we're working on different type of linkers, really developing these type of linkers in-house, so to have optimal exposure in the tumor tissue. And with that, I think with Trypic, there are multiple opportunities for different type of APIs, small molecules as well as also peptides, to address the current challenges and to develop products with a therapeutic profile. And with that, I'd like to thank all the different patients who participated, but particularly also the collaborators and the investors, and clearly also the team in-house. Thank you for your attention.
Thank you, Cristianne. I will open it up for questions. I think I'm waiting for questions to come in. In the meantime, Cristianne, what is the intent with the docetaxel at present? I think you said there was a crossover trial and then a possible phase 2. What indication are you thinking of pursuing?
So currently, the docetaxel is being evaluated in ovarian cancer. That's a small study in which we're trying to evaluate both the safety as well as the efficacy profile. That is currently being analyzed, meaning the recruitment is now finalized. So we're currently analyzing the data. And based on those results, we take a look going forward, either in ovarian cancer or maybe in an indication in which Tageted is already registered. So you could think of breast cancer, lung cancer, or prostate cancer.
Okay, and in terms of the efficacy, I think you said in phase 1 you saw some partial responses. Can you speak a little bit about that and which type of tumors that was seen?
Yeah, well, clearly a phase 1 study is a study with all comers, so we could not really identify certain, let's say, patient population or indications. But we particularly saw some patients with drop in the PSA, so prostate cancer, as well as also a patient with ovarian cancer, where we saw also a drop in the CA-125 level. So particularly biomarker response, we saw based on RECIST, we saw also some shrinkage, but not formally according to the RECIST criteria.
Okay. Any other questions from the audience? Okay, I have one follow-up question. In that crossover study, you mentioned that the drug level in the tumor was higher, but I couldn't quite catch it. But over a longer period of time, you were seeing the same drug level. Could you just explain that or clarify?
Yeah, sure. So what we gave to this patient is Tageted or a nanoparticle, but we actually saw up to 2 weeks time. I think it's particularly visualized by the light green bar, but particularly the entire period of 2 weeks time, Tageted remained quite stable, constant, actually unexpected high levels, meaning there was no decline that we expected. We expected to have an increase of the levels in the tumor tissue after administration, followed by some decline up to a couple of, I don't know, days, but surprisingly up to 2 weeks time, we had fairly consistent levels of the Tageted over time.
Yeah, and that was comparable to your Cripex as well?
That's correct, yeah. So that's why we could not show any statistical significant difference between the Tageted levels or the released material coming from our nanoparticles.
Okay.
May I ask a question?
Yeah, please go ahead.
Okay, I'm Bart. Hello. I'm taking part in this session. Okay, Cristianne, wonderful. I have a question, two small questions actually. One about the distribution of the Cripex in patients in blood. I think it was done 2 days after infusion, and you see a very spotted distribution over on the top left panel. You see in the vasculature, that's at least my impression, a very spotted distribution, very focal, very localized in the vasculature, the big vessels. That's my first minor question. What is that?
Well, I couldn't really judge if it's truly focused, but rather clearly after 2 days, there is still a lot of material circulating in the bloodstream. We know the nanoparticles are really confined to the blood compartment, so it's a fairly dense profile that we see still in the bloodstream.
Okay, but you see these spots there, these black spots throughout the vasculature. What is that? If you go down in the middle of the body, over there, yes. What is that?
To be honest, I would have to check with the radiologist because I'm not in depth acquainted with how you should interpret these data.
Okay, because it reminds me of like what we've seen with cardiovascular patients where there was uptake of particles in the plaques, atherosclerotic plaques. But this was totally different, of course, here. It doesn't have anything to do with it, but it's just that it caught my attention. And I have another question. In the other picture, in the slide after this slide, you see, I tend to sort of see that the localization of the Cripex is different than the FDG PET. It's not exactly co-localized. So how is the distribution of the particles in the tumor as compared to the tumor tissue?
Well, that's absolutely a very fair point. What we see is indeed there is uptake in the overall tumor lesion, but it's clearly a spotted pattern, meaning it's absolutely not nicely homogeneously spread. So it's clearly one of our observations that not all tumor lesions are being positive for the zirconium particles, but even if they're positive, it's clearly very scattered. So that's something that we absolutely have to further sort out, meaning what can we do to improve the tumor uptake, but even more importantly, what can we do to make it more nicely spread homogeneously throughout the tumor tissue?
Okay, thank you.
Thank you.
All right, thanks. We need to move on to the next presentation. Thank you, Cristianne.
Thank you.
The next presentation will be by myself. I'm Dr. Neil Desai from Aadi Bioscience. I will be presenting on a registration trial of Navcerolimus in malignant PEComa. So I'm going to share my screen here. Can everyone see the screen? Okay. All right. So thank you for being here today. It's my pleasure to present some data that we have from the clinic on Navcerolimus. So what is Navcerolimus? I think most people are familiar with Abraxane and the technology behind Abraxane, which includes taking albumin, human albumin, combining it with a drug. In the case of Abraxane, it was with paclitaxel. In the case of Navcerolimus, it is the mTOR inhibitor sirolimus. So we create these nanoparticles in a special technique, combining with the protein, and then you have protein-bound drug as well. This is now well described, the mechanism of transport through the vasculature using the caveolar transport and albumin receptors on the endothelial cells. So what this gets us is we get high tumor accumulation because we are leveraging the albumin pathway and accumulation in tumors of albumin. It changes the PK profile, so longer half-life, AUC exposure, etc. We have significantly improved therapeutic index and safety profile, and we see increased target suppression of the tumor. And this is particularly important where you have to hit certain targets fairly hard to get activity. So this has been proven with Abraxane to show good effectiveness in different tumor types. So the focus of this drug, Navcerolimus, or ABI-009, is really focusing in on the mTOR pathway. So mTOR, I think most people are familiar with, is a ubiquitous pathway that results when it's activated in proliferation, change in glycolysis. It's also involved in autophagy, inflammation, and regulation of T cells and immunity. So it has many different functions, and therefore, as you can imagine, it may be relevant to many different diseases. What we're focused on is looking at certain mutations in this pathway that result in activation of mTOR and therefore result in the case of cancer, proliferation of the cells. So the first indication of interest is advanced malignant PEComa, and we'll talk more about that in the upcoming slides. We're also looking at specific tumor mutation-driven studies in patients with TSC1 or TSC2 mutations, and I'll show you a little brief data on that, more recent data. We have a trial ongoing in metastatic colorectal cancer that focuses in on mutations in PTEN, and also glioblastoma, which is well known to have aberrations in PTEN. We can go outside of the cancer field as well, because the mTOR pathway affects not only proliferation, but glycolysis and autophagy, etc., which are important in pulmonary hypertension. In the pediatric setting, in refractory epilepsy, there's often mTOR activation in different parts of the brain that result in epilepsy. And so we're looking at pediatric studies in this indication. We have another interesting study about to start in mitochondrial disease, which is also affected through the mTOR pathway and glycolysis. So that's another interesting indication. And last but not the least, because of the strong immune modulation of sirolimus, we can also look at it in COVID-19 ARDS. So the way we are developing this, firstly in cancer, is looking at a very orphan indication, which is PEComa. It's a very rare disease. There's probably 300, 500 patients in the US and in Europe, similarly. And this is often driven by mutations in the mTOR pathway through TSC1 or TSC2. And then beyond that, we go in broader than PEComa, outside PEComa, to multiple different cancer types, all of which may have TSC1 or TSC2 mutations, colorectal cancer with PTEN mutations, and numerous other indications, both in and outside of cancer. So what does this do in the preclinical setting? And we've looked at tumor distribution of the drug, and compared it to other inhibitors of mTOR with like Sirolimus and Everolimus, which are known drugs given by the oral route. And here at the same dose, we get significantly higher levels of drug in the tumor. And depending on what you're comparing against, between 12 to 43-fold higher drug levels when you're given the same total weekly dose in tumor-bearing animals. So this is in an aggressive bladder cancer model. And we also looked at target suppression of PS6, which is a downstream activator of mTOR. So by doing immunohistochemistry, you can determine how much suppression of mTOR you have. And even at the low dose of our drug, Navcerolimus or ABI-09, you see very efficient target suppression in the tumor compared to what you may see with the other drugs, Sirolimus or Everolimus, which look more similar to the control. And correspondingly, in the same animals, if you measure the tumor growth, you can see that you get significantly higher tumor suppression as a result of the higher drug levels and better outcomes. So this clearly shows that the level of drug going into the tumor is important. We did also, in this study, we pushed the dose of oral Sirolimus and Everolimus to 6-fold higher than what you would normally use in the clinic. And it took about 6-fold higher dose to try and approach the levels of target suppression and tumor suppression to get compared to ABI-09 to reach that level of activity. Of course, you can't do this in the clinic because of toxicity issues, but this proves the point that the amount of drug getting to the tumor and the activity in the tumor is very important, and that the Navcerolimus allows you to do that. So we went into the clinical study with malignant PEComa. PEComa is a very rare form of a sarcoma, and it has distinctive cells that show focal association mainly with blood vessel walls. So it has no specific organ of origin. It can be originate anywhere in the body, and it can be very aggressive in the malignant phenotype. And as I mentioned, there's about 300 to 500 patients annually in the US who develop PEComa, so new cases. And cytotoxic chemotherapy, which is commonly used in sarcoma, does not work at all in this disease. And there's no drugs today approved specifically for the treatment of PEComa. Now, there's been some case reports of mTOR pathway activation in PEComa, and the use of mTOR inhibitors has shown clinical benefit. And this was based on some of these case reports. We decided to do a study with our drug in these patients. And as I also mentioned, the mutations in this pathway, mTOR pathway, especially TSC1 and TSC2, are predominant in this population. So here's the design of the study. We went to the FDA and had a discussion with them about the appropriate study design to get the drug approved in this setting. And because it's such a rare disease, we agreed upon a small study, a phase 2 study of about 30 patients. And the endpoint was response rate for approval, independently reviewed response rate. And the target was about a 30% response rate in about 30 patients. And so if we hit that target, then we meet the primary endpoint. So Navcerolimus was given at 100 milligram per meter square on day 1 and 8 of a 3-week cycle. And then we keep treating the patients until they progress or may have toxicity. So this is the demographics of the study. We enrolled 34 patients and predominantly women. There's 82% women. This disease is known to occur more commonly in women. And metastatic patients were 85% of the population, and locally advanced and inoperable were about 15%. And you see that the disease site where the primary disease originates is varied. About 25% in the uterus, 18% in the pelvis, retroperitoneum, also in the lung, kidney, and even in the brain, etc. And we had patients with metastases. About 70% had lung metastases, 21 liver metastases, and then others. So as far as the safety profile, the drug was well tolerated, and we had only 2 patients discontinue for toxicity out of the 34, but the rest of them tolerated the drug pretty well. As far as hematologic adverse events, they were fairly mild and well controlled. We had some anemia and thrombocytopenia. We did see stomatitis and mucositis in about 79% of the patients. However, this was also well managed. It occurred early in the treatment, but could be easily managed with things like steroid mouthwash, etc. And then we had some rash, some metabolic disturbances, which are well known for mTOR inhibitors, etc. But no new events or no unexpected adverse events. We did see some pneumonitis, which is also known with mTOR inhibitors, but it was only grade 1 and 2, and it was reversible. So moving on to efficacy, this is a waterfall plot showing the results. And here you can see that we saw a very nice response in these patients. And the overall objective response rate was 39% by independent review and 42% by investigator review. So it was well matched. And then we had stable disease in about 50% of patients and progressive disease in 10%. What was also interesting that in a particular histology of uterine PEComa, which is known to be difficult to treat, we also saw a 43% response rate, which was similar to the overall population. And the responding patients were not restricted to a particular region, but they had responses in the uterus, kidney, retroperitoneal, pelvis, etc. So here's some examples of some CT scans of those patients. So these are a couple of patients with uterine or primary, and you can see the metastases and then disappearance of the tumor. Here's another patient with a primary site of uterus, metastatic to pelvis and lung, and then good response. We have a patient who had retroperitoneal primary and had metastases to the liver, and the lesions disappeared. Patient with metastases to the lung and very good response in the lung as well. And also from a primary side of a kidney, we had a large lesion that had a very good shrinkage. So the patients tolerated the drug well, stayed on treatment for long periods of time, and you can see the individual patient graphs of the tumor size versus time. And the axis down at the bottom is in weeks. So, 52 weeks is a year, and you can see that several patients are ongoing for more than 2 years and 3 years. The arrows indicate patients that are still on the study. The striking thing is that once the tumor shrinks, to a response, then we see the continued maintenance of that response over long periods of time. We had one patient that achieved a complete response on study as well. So the duration of response, the median has not been reached, and the calculated median right now is at 25 months and ongoing, as several patients are still on therapy. We also did a mutational analysis on these patients. So this is the same waterfall plot. What's very interesting is to look at the patient tumor regression by mutation. And we see the TSC2 mutation is indicated in the orange color. And so a lot of the patients that had significant tumor shrinkage, in fact, had the TSC2 mutation. And when we did the correlation, out of 9 patients that had TSC2 mutations, we had a response rate of 89%. So 8 of those 9 patients responded. And in fact, the 9th patient also had a response, but we didn't have a confirmatory scan, so it was called a stable disease. So essentially complete response in the TSC2 mutation data set. We also looked at phospho-S6 as a marker, and we saw the correlation of those patients who were positive for phospho-S6 also did very well on the study. we've done mutational analysis across looking at many different genes. So I won't get into the details, but really the TSC2 was the really only significant correlation that we saw besides the PS6. And responses, as I mentioned, occurred across many different sites of the tumor. So in summary, the registration trial met its primary endpoint. The independently assessed objective response rate was 39%, which included one patient with a complete response. The responses were highly durable, and the median DOR was not reached, and 50% of the patients with a response exceeded 25.2 months and are still ongoing. The patients with TSC2 mutations had an 89% response rate, so very high. So this is a very interesting signal that we're following up on. And so this is the first prospective study in advanced malignant PEComa, and suggests that Navcerolimus is highly active for this aggressive sarcoma for which there are currently no approved therapies. And we're planning a TSC1, TSC2 pan-tumor agnostic study. So I'll show you some early data on that. So here's data now outside of PEComa. So this is a patient with a high-grade sarcoma. Happens to have a TSC2 mutation and has been treated with chemotherapy, adriamycin, ifosfamide, gemcitabine, taxotere, pazopanib, pembrolizumab, and then failed all of these therapies. And you can see the PET scan with the tumor when the patient came on our study. And then within 6 weeks of treatment, so essentially only 2 cycles of treatment, the patient had a near complete response. Most of these lesions are gone. So there's fairly dramatic response in this patient. We have another patient with metastatic angiosarcoma. This patient has a TSC1 mutation and had a primary tumor in the heart. It's a right atrium, and also metastases in the lungs. So this is the pictures on the left are at baseline. You can see extensive metastases in the lungs and a large tumor on the heart, which was not responsive to doxorubicin, ifosfamide, mesna, and Taxol. And then at 5 weeks after therapy, we saw quite a good shrinkage in the atrial mass and also disappearance of several lesions in the lung. So this is, of course, early data from just a couple of patients, but we're following this up with more studies in patients with TSC1 and TSC2 mutations. So beyond this, we have a study ongoing in colorectal cancer with patients with PTEN loss. We're also looking at pulmonary hypertension, which is a very interesting disease, and mTOR is a driver in that disease. We have surgically refractory epilepsy. This is in pediatric patients. The study is ongoing. It's a phase 1b study. And then we're planning to initiate a study in Lee syndrome, which is a mitochondrial disease that we will initiate later this year. So with that, I'll stop and thank you, of course, to all the patients and the families and to the attendees of this conference. I'm happy to answer any questions. Okay. Any questions?
Maybe I can ask a question, Neil. This is Cristianne. I was just wondering, because you now gave the Sirolimus intravenously, and the comparator is oral, right? Meaning that the bioavailability of oral Sirolimus is probably significantly less. So is it a fair comparison in sense of what the extent of tumor uptake is?
Yeah, so we can push we do it at the relevant clinical dose, right? So if it's oral Sirolimus or everolimus, they have a range of dose that is given in the clinic and approved. So that's the dose that we test at, because the therapeutic window for those drugs is very narrow. And that's a problem with those drugs today. and so you're limited to whatever the clinical dose is. Okay. All right, I'm looking if there's any questions from the audience. Let's see. Okay, so let's move on. I'll thank myself and we can move on to the next speaker. The next speaker is, let me just get here, is Stefan Albert. Stefan, I see you on the screen. So Stefan's going to talk about Talidox. It's the world's smallest liposomal doxorubicin, and give us his insights from the first applications in patients and immuno-oncological potential. And Stefan is from Innomedica in Bern. Go ahead, Stefan.
Hello, good morning, everyone. Thank you very much, Neil. It's my pleasure to present to you our learnings about making doxorubicin liposomal and changing its properties to beneficially influence the distribution and efficacy of it. Our slide is not switching. Yeah, so basically we set off to address three distinct needs. On one hand, of course, you want to penetrate the tumor tissue as good as possible and let the drug be retained within the tumor. As well, you want to modulate probably the tumor microenvironment in a way that it becomes hot immunologically. So you want to provoke immunogenic cell death and migration of T cells, etc. And of course, you don't want to have the nanomedicine to have any unnecessary events provoked. So the design rationale is fairly straightforward. We built on the success that the Calyx has had for the last, I would say, 25 years. So we thought we use probably the same mechanism of crystallization, but with slightly different tweaks, and all these tweaks start up to add on to each other. So, of course, particle size has been discussed a lot, and it is highly influential in how the drug behaves in vivo. One beneficial aspect of making it really small is that actually the EPR, or maybe even the transendothelial location that is currently now being discussed, is more prominent when the particles are really small. Also, of course, the cellular uptake is faster when particles are really small, as well as the diffusion speed of smaller entities is faster, and hence extra tumoral matrices can be diffused to a faster, better extent. As well, we tweaked the drug release mechanism in a way that we crystallize the drug less dense, so there is a faster release when the liposome actually disrupts. And we tried to maximize the particle density within the formulation, so we greatly changed the drug to lipid ratio actually in the opposite direction, to have a lot of lipids and to have rather few drug. Because we thought if the nano system really does change the behavior, then you probably want to have a lot, or the most amount of the nano system injected into the body to have the maximum effect from the nano system, while you want to minimize probably the drug density within the liposomes. So that adds up to a significant drug-to-body interface of a couple of 10 to the power of 17 square nanometers, which is about 10% of a square meter. So the drug interacts with this interface with the body. And in a head-to-head comparison, you can see here to the comparator that the particle density is about 3 times higher, while the particle size is about half of it. And the drug concentration is also about half of it. So we have 1 milligram per milliliter doxorubicin, while Calyx is formulated as 2. So that ends up in the positive impact of drug accumulation in tumors over time, which we have seen here in one of many cancers we have tested, but I'm showing here the A27 ovarian cancer model. And you can really see that over time, drug is accumulating faster with Talidox formula, and while liver clearance, interestingly, is the opposite way around. So it's rather lower. So that is ending up in a, I would say, rather positive tumor to liver ratio. So, of course, the preclinical models are only limited in predictability on what happens in men. And we went on to make a phase 1 study that is currently still ongoing. And we have amended the protocol several times to include further patients, since the drug was doing really good. So in a nutshell, we conducted the study with the SAKK, so the Swiss Group for Clinical Cancer Research. It started off to find the MTD, which was not really reached, but we stopped at 45 milligrams per square meter every 3 weeks, because sooner or later there were toxicities occurring after repeated cycles. So we thought like there is not a need to go higher at the current dosing frequency. Included were patients with advanced solid tumors of different origins, and they could have had a maximum of 3 prior lines of systemic chemo. And here I can present you some limited findings that date as of April, so there is more to it, but due to confidentiality issues, I cannot present more advanced data yet. But interestingly enough, we saw full dose linearity and a very, very long serum half-life of 98 hours, which was rather surprising to us, because in the murine models, we always saw a rather short half-life. And interestingly, in humans, it doesn't seem to be the same as in the rodents. So Talidox really has one of the longest half-lives of nanomedicines I have seen so far. And I think this is really interesting because if you want to accumulate in tumors, I think being available in the bloodstream is one very important prerequisite to sort of specifically interact with the tumor tissue over time. So from these 13 patients, in terms of efficacy, I can tell you these are here we include also the very low dosing levels that we started off with 10 milligrams per square meter, which was probably very mild dose, but could have had some therapeutic effects. So it was our starting dose, but of course there's some dose response relationship. But interestingly, there in these heavily pretreated patients, we saw clinical benefit in more than half of the patients at that dose level, including PRs, and there have been more PRs now when we were including more patients. And I think, of course, efficacy is difficult to discuss at that point. And what we can do is to look at literature data regarding side effects, since these patients have had already a number of cycles administered and are now fairly advanced in the sample size. And it was interesting to see that side effects such as vomiting, nausea, and alopecia, as well as myelosuppression and neutropenia, were very, very low as compared to the compendial data we took as a comparator level for the Calyx formulation. So that is a positive thing we hope to build on. There is now a second study amendment coming up that we call the SMARTER. It's an acronym for that Talidox is actually smaller and it can be dosed a little bit in a stronger intensity, so that that ends up. And it will be a intra-patient crossover design to compare the PK values of the Calyx formula against Talidox in the same individuals with a 12-month follow-up post-study, and we'll measure the pharmacokinetics as well as tolerability and response, of course. As well, there are now intense discussions going on about the possible phase 3 scenarios, whether we should benchmark against the free drug paclitaxel or nab-paclitaxel, or against Calyx, or just leave it open and go for the investigator's choice. Most likely, our target indication will be metastatic breast cancer, first line or second line or any line, we'll see. And of course, that would be the most simple thing to compare monotherapy against monotherapy, but currently I think it's also becoming very clear that immunotherapy and chemotherapy are just very well in synergy, and we should also make efforts to combine those. So I want to quickly talk about the combination of immune checkpoint blockade with Talidox, and just mention that we do also have an imaging-based approach to have a more rational inclusion of patients when to treat with Talidox. But I'll focus now on the immunological aspects. So it was interesting to see that Talidox upregulates the marker of immunogenic cell death, HMGB1, to a greater extent as compared to Calyx, as well as the calreticulin marker was upregulated to a higher extent at basically all those ranges. So we think this is rather ideal, and of course this was a cell culture system and just shows the cellular uptake and induction of these markers on the surface of these cells. And the biggest question is, of course, how are these markers induced in vivo in real time? And we don't really know that yet, but there are some pieces of evidence that line up together, and I want to quickly outline that here. So one of the most interesting combinatorial studies have been the TONIC trial by Vorwerk, published last year, where they showed that Doxorubicin might have been one of the best agents to combine with PD checkpoint blockade in triple-negative breast cancer, so an indication of high unmet medical need, where other chemos sort of didn't do as well. So that was, of course, with the free drug formula, to my best understanding. And we went on and to see if in the preclinical models we can do something that is in that direction. And we found that if you treat the CT26 colon carcinoma in mice with Talidox, you basically shrink the tumor to inexistence, and it doesn't regrow anymore if you stop treating. But it was interesting then if you have a second tumor injected into the opposite flank of the tumor of the mice, you see that the immunological memory and the response of the host against the second tumor challenge is much bigger when the first tumor is fought against when PD-1 checkpoint blockade was applied. So immunological memory is highly positively influenced by anti-PD-1 combinations, and we basically anticipate that Talidox can, to a very high degree, induce HMGB1 and calreticulin in tumors, while PD-1 influences the T cell memory response during cell death, and that ends up in an improved immunological memory and might end up in a beneficial therapeutic outcome for patients sooner or later. So we'll be looking into that in the near future. We manufacture Talidox in our own facilities in Switzerland. If you want to see our facilities, you can come by for a visit. The door is open. And we do have a pretty broad product pipeline. Besides that, we have diagnostic agents coming up with near infrared imaging, and we have a very promising drug in Parkinson's disease as well as Alzheimer's disease that will be presented later on today. So summing up, Talidox is one of the smallest, if not the smallest, liposome on the market or in clinical testing, as I know. It has a very low drug-to-lipid ratio that is beneficial in terms of a low liver, high tumor uptake profile, as well as cellular uptake. It has a very long serum half-life, and it has been shown that serum half-life might correlate with the longer the serum half-life, the better the therapeutic outcome for liposomal drugs. I think Mr. Barnholtz showed that lately for his drug Prometheal, and we didn't look at. We'll certainly see if our own data will show the same, but we'll need more patients to make these kind of conclusions. Currently, the tolerability profile of Talidox is highly beneficial with reduced myelosuppression, neutropenia, and nausea and vomiting also being reduced. There's a strong induction of immunogenic cell death with HMGB1 and calreticulin. And I think Talidox is positioning itself as an interesting combinatorial agent for checkpoint blockade on the long term, because it exposes the tumor to a high amount of drug, as well as causing doing that over a very long period of time. But certainly these data are not conclusive, but well, I think currently we feel that it's going into the right direction. So we do have also financing rounds going on, and if you're interested, do get in contact with us. Thank you very much for your attention. If there's any question, I'd be happy to take them.
Thank you, Stefan. Any questions from the group here? I'm monitoring the question. Well,
yeah, Neil, I have a question. Stefan, thanks for a wonderful overview. Great results. Interesting also, such a small liposome product. You mentioned in the beginning that you aimed at very loosely packing the doxorubicin in the liposome. Were you able eventually to find out if that improves bioavailability, if that helps the product work better?
Well, if you load too much doxorubicin into the liposome, the crystal grows really large and the liposomes get deformed, which I think is maybe not ideal. Energetically speaking, the liposomes should probably be in a minimal energetic position, which would be the spherical round form. So that is one aspect. If you overload the vesicles, they become like these coffee beans. And on the other hand, there's just a clear correlation. The higher you load the drug, the slower the release of the drug at the end, because the crystal becomes just more and more dense. So we try to find a sweet spot to still get the therapeutic amounts into the drug, unless at the end, if you play the game into infinity, you would have to inject 1 liter of liposomes into a patient, which I guess is not possible. Currently, the administered volume that we inject is about 100 milliliters. And I think we could again split the lipid to drug ratio downwards to inject at the end maybe 200 to 300 milliliters of liposome product at the end. To go further down there and to see if this would again increase the tumor uptake and the drug release.
Okay, and if you inject way more lipids, don't you get into issues with regard to infusion reactions? Did you have issues with that?
No, nothing. I think we knew from calyx administrations that some 8 milligrams of dexamethasone just make infusion reactions disappear. So that was never an issue. But it might, if you increase the amount of injected drug, that might common issue sooner or later, yes. But generally, I think the liposomes are ideal because they themselves are not toxic at all. So I think you can inject really large amounts. I mean, people that get parenteral total nutrition, they get injected with lots of phospholipids just for as a nutrient purpose. So I think.
Stefan, there's a question from Gert Storm,
Yeah.
and the question is skin toxicity dose limiting, or do you see hand-foot syndrome?
Yes. Yes, thank you very much. I wanted to touch on that. Yeah, that was actually a very big surprise because in the GLP toxicology assessment, as well as in mouse studies, we found a complete absence of hand-foot syndrome, while in the human study, we did find skin toxicity that occurred, and we had to it was one of the dose limiting factors, or if not, it was the dose limiting factor. And it really came as a surprise because we didn't see it at all in the preclinical testing. So that was very interesting to see how the two species really compare also.
Yeah. So I have a follow-up question on that, which could be related to size. So the hand-foot syndrome, what is the differences? Are you able to tell between calyx and what you're seeing in your case?
No, I think it's fairly comparable. The rate of hand-foot syndrome, it might be a little bit lower for Talidox, but we don't really know yet. We went for a 3-week dosing cycle for the phase 1, which makes also the direct comparability of data quite difficult. We went for the 3-week dosing because we saw that the drug concentration in blood really is basically 0 after 3 weeks. And there's no carryover effect if you start redosing at 3 weeks. So we went for the 3 weeks. And it depends. I think hand-foot syndrome becomes more prominent if you increase the dose and the frequency at the same time. So there is this limiting toxicity that we must respect, yes. But if you go for the current dosing strengths, that is done a lot in the clinics, is 10 milligrams per week, given in a 4-week treatment regimen, so 40 milligrams every 4 weeks. And if we do the very same with Talidox, so 30 milligrams every 3 weeks, which is the same strength, then we have a very, very low occurrence of hand-foot syndrome, as well as any other toxicological reaction. So I think at the end, it might end up that because of the long serum half-life, the body has a long time to metabolize all the doxorubicin that has been injected and all the lipids. And that gives just a smoother profile at the end. Okay.
All right, thank you, Stefan. We'll move on to the next speaker. And thanks. So the next speaker is Gilles De Vita, and he's the CEO of Divincel SAS in Montpellier, and also works with Aadi in the US. And he's going to talk about novel peptide oligonucleotide complexes for mRNA and gene editing therapeutics. Gilles? Gilles, you're on mute.
Is it okay now?
Yes, we can hear you.
Yeah. Okay. So, good morning, everyone. I'm very pleased today to present our technology. So we are developing a peptide-based technology. It's mainly, I changed the title of my talk. It's now I'm mainly today going to talk about peptide-based nanoparticle for KRAS oncogene mutant gene editing, specifically in tumor cells. So our main target is KRAS. You may all know KRAS. So it's one of the major oncogenes and is playing major role in cancers. So KRAS is mutated in more than 40% of human cancers, and it's involved in several pathways and control cell survival, proliferation, cytokine secretions. The interesting point about KRAS, mutation is on KRAS is always single base missense mutations. It's more than 90% of the case. And there's three main residues is G12, G13, and Q61. And if we analyze in detail what's happening in pancreatic and colorectal cancers, the main mutation is on the G12 codon. And if you zoom on the G12 codons, the mutation is mainly on G12D and G12V. And so far, there is no inhibitors and direct inhibitors of those KRAS mutations. And what we are developing, what we are developing at Aadigen and Diving Cell, it's a technology called DiveTrex, which specifically target KRAS mutations at the codon 12. It's the strategy is to combine CRISPR, so gene editing technology, with a specific gene editing of the two type of mutations, the G12D and G12V. And we combine them together with the tumor-specific nanocarriers we have developed at Diving Cells, which is able to directly target the tumors. I'm not going to go in detail on CRISPR. You may all know that using CRISPR technology, we can modify the gene, we can add some correction at the genetic level. What is important is the CRISPR-Cas9 machinery is involving a protein, Cas9, it's a RNA guide nuclease, together with a single synthetic guide RNA. So we have two components there, and we have to get those two components inside the cell and in vivo. And that's one of the major limitations right now for the CRISPR-Cas9. It's the delivery. So what we are developing in Aadigen, it's a technology we call AadiGN. It's a peptide-based platform, which mainly works for the delivery of nucleic acid, but also for other components like protein and small molecule. This technology is based on amphipathic peptide that combine both electrostatic and hydrophobic interactions there and there. And that, with that, we can make some interaction with the nucleic acids and interaction with the cell membrane. One of the advantages of those peptides, we can add at the C terminus some different motif for the stabilization and for the targeting. So those peptides can form small molecules, small nanoparticles in a complex with nucleic acids. We call the AadiGN nanoparticles. And specifically for tumor targeting, we have developed a specific peptide, which is called DiveTrex. DiveTrex contains the same structure as the AadiGN peptide, but at the N terminus, we have had a tumor targeting motif. It's a small motif, which is able to target the tumor. And those peptides are forming also a nanoparticle with nucleic acid. And those nanoparticles have at the surface this targeting motif. So what we show that this peptide are able to form very stable nanoparticle with nucleic acid, small size or large size. That's one of the main advantage. We can form nanoparticle with mRNA or Cas9 and guide RNA. The nanoparticles are small, between 50 to 100 nanometers diameters. The charge is close to neutral. We can freeze dry the particles, they remain stable, and they remain stable for more than 12 months in parallel. What's the advantage of the particle is promote very high efficiency in terms of delivery of the CRISPR machinery inside the cells. The mechanism is also one nice property of the particle. It's not going through the endosome. We have the property of the particle are able to interact with the cell membrane, and that way to increase membrane fluidity, and that way the particle can enter the cells. We didn't see any major toxicity of those particles in vitro. And for gene editing, we got very good, robust gene editing in several cell lines, including very sensitive cell lines. So our goal was to be able to target specifically the tumor. So I show you that we have the DiveTrex, so with the tumor targeting motif. And what we show is this DiveTrex is able to deliver mRNA directly inside the tumor. So the strategy present here, we just get two types of mice with two types of tumors, human pancreatic tumors, the PANC1 tumors, or mouse breast cancer tumor, 41. And we inject IV the Dive-Tregs complex with a Cas9 mRNA or luciferase mRNA. And we look first for luciferase expression in vivo. And as shown on that panel, if we look on the human panc tumors, when we have Dive-Tregs, we show very nice delivery of the mRNA inside the tumor, but also a nice expression of the mRNA in the tumor, which is not the case with the Aadi gene, so without the targeting motif. And in that case, we have luciferase expression in the lung and in the liver. And it's the same thing for the other type of tumors, it's the mouse breast cancer tumors. So we have very specific tumor targeting, and there's a specific work in a human tumor and mouse tumor. We also show that using this Dive-Tregs approach, we can increase the delivery of mRNA Cas9 inside the tumor. So on that graph, we test several conditions, and basically you have to focus on the red one, and the red one is the Dive-Tregs. And when we use Dive-Tregs, we have a very strong expressions of Cas9 inside the tumor. So we can really get specific Cas9 expression inside the tumor. So we, as I said, we combined specific CRISPR guide RNA to target specific mutation of KRAS together with the tumor targeting. So we have selected three highly specific guide RNA which target different mutation of KRAS. Those three guide RNA were evaluated on several tumor cells. For PANC1, pancreatic cancers with a G12D mutation. For SW403 is colorectal cancers for G12V mutations. And we did some work also on lung cancer cells with a G12V mutation, G12C mutations. And as control, we use HT29, which is colorectal cancers, but with wild-type KRAS. And we evaluate Dive-Tregs on those cell lines. And what we find is we have a very, very selective gene editing, which is we got more than 75% editing at the codon 12 mutations. And as you can see here on the PANC1, only the G12D guide RNA worked with more than 75% gene editing, and similarly on the other type of tumors. We show that knocking down KRAS mutant block cancer cell proliferation, and this, in that case, is also highly specific to the guide RNA we used. And so we can block cancer cell proliferations, and we didn't see any toxicity on the cellular level of our complex. We compare it to lipid-based nanoparticle. So we then evaluate the Dive-Tregs KRAS target system in vivo, first on pancreatic tumors. The experiment was designed as the following: we inject the pancreatic cancer cells in the mice, and then we let the tumor to establish for 2 weeks, and then we start the treatment. And the treatment was basically 2 injections at day 1 and day 7 of the complex Dive-Tregs with a CRISPR editing of KRAS G12D. And then we look for tumor growth, and basically as reported on the tumor growth graph, we use as a control a saline solution, so we have no effect. But the other control was also using a non-specific guide RNA, which target another mutations of KRAS, which is not present on in the PANC1 cells. And so there were no effect. And if we use 0.5 mg per kg of our Dive-Tregs specifically targeting G12D, we block by 50% the tumor growth. But with 2 treatments, 1 mg per kg, we completely block the tumor growth. We even see the tumor shrinking. The other interesting point is we didn't see any effect on the body weight, no body weight loss. So the treatment were pretty well correlated. We validated that this effect on the tumor growth is directly due to the fact that we are editing KRAS G12D. For that, we analyze the tumor after 50 days at 2 levels. We look for KRAS G12D gene editing, and we look also for the level of downstream signaling proteins. As you can see here on the gene editing, we clearly have a strong gene editing of KRAS G12D inside the tumor by more than 80% in the group 4, which is 1 mg per kg. And if we look at the KRAS downstream pathway, we look for AKT and AKT phosphorylations at 1 mg per kg. We have a very strong down-regulation of the phosphorylations. So the effect is really clearly specific to KRAS G12D. What about the other mutation, G12V? We test our Dive-Tregs on KRAS G12V, this time on colorectal cancers. In terms of protocol, it was basically the same one than on the PANC1. We use human colorectal cancer cells. We inject the cell, we wait for 2 weeks for the tumor to be established, and then the treatment starts by only 2 injections at day 1 and day 7 of our complex. We look at the tumor growth. The control had no effect, even with a missense G12C guide RNA. When we use our specific G12V guide RNA, 0.5 mg per kg, we block by 50% the tumor growth. 1 mg per kg, we completely abolish the tumor growth and even see the tumor shrinking. So two injections were enough. We didn't see any tumor emergence after 80 days, after the starting of the treatment. And all the treatment were well correlated. As for the PANC1, we show that the effect on the tumor growth on the colorectal tumors is very specific to the fact that we have KRAS G12V editing. If we analyze the tumor after 50 days, we can see that KRAS G12V level is reduced by 85%. And if we analyze the downstream pathway, AKT and AKT phosphorylations on group 4, with 1 mg per kg, we reduce the phosphorylation of those 2 proteins by more than 60 to 80%. So it's very highly specific mechanism. What about the toxicity? We look for several markers for hepatic and renal toxicity in vivo, and in all the cases, whatever the group we use, we didn't see any significant alteration at the kidney and liver functions. This was 1 mg per kg, but we went up to 5 mg per kg and didn't see any difference. It's not shown here, but we didn't see any change in the organ index and no significant cytokine activations. An important point in the CRISPR field is the off-target. So we checked previously that at the cellular level, we didn't see any off-target. And then we check in vivo if we can see any off-target or modification of housekeeping gene. So for that, we analyzed 3 housekeeping genes in the different organs and in the tumor with a different group of treatments. And in all the cases, either GAPDH or HPR1 or MAITP6, we didn't see any change in the housekeeping gene expressions. So there was no side effect linked to off-target or downregulation of housekeeping gene associated. One important point of our technology was to be able to combine it with other drugs which are already used on the patients, and to check if we have any synergy between our technology and other drugs. So what we showed on this experiment, and we combine our Dive-Tregs technology with a pancreatic, with Abraxane on pancreatic cancers. So the experiment was set up like this way. We inject the PEComa, wait for 2 weeks, and start the treatment. Treatment was as previously 2 injections of our Daphtirex nanoparticle at day 1 and day 7, and we inject weekly the Abraxam at 50 micrograms. What we showed is that's a control, but if you look at the green curves, is the Abraxam, so we block tumor growth by 50%. Our Daphtirex 0.5 mg per kg is similar, 40-50%. If we combine the two, 50 microgram Abraxam, 0.5 mg per kg Daphtirex, we completely abolish the tumor growth. So there's clearly a synergy in vivo of our Daphtirex technology with the current method used like Abraxam for the treatment of pancreatic cancers. Similarly, for colorectal cancers, we combine Daphtirex targeting KRAS mutation G12V. We combine with capecitabine. We have the same type of protocol. We wait the tumor growth for 2 weeks. Two injection of our Daphtirex at day 1 and day 7, an injection of capecitabine weekly, 20 micrograms. We analyze the tumor growth, and what we showed is that the control in red, it's our Daphtirex 0.5 mg per kg. It's around 50-40%. Similarly, with capecitabine, we combine the two at the same concentrations. We completely abolish the tumor growth. So there is also a synergy of our technology with other drugs which are already used for the treatment of colorectal cancers. So to conclude on Daphtirex, so we have developed this technology which combine highly specific CRISPR approach using highly specific guide RNA with the tumor targeting nanoparticle. With Daphtirex, we show that we can induce permanent and specific KRAS G12V and G12D gene editing in vivo. It's really specific to KRAS mutations, and we show that we can block downstream pathway in vivo, abolish both colorectal and pancreatic tumor growth in vivo, and now we are working also on lung tumors. This approach can be used in synergy with the standard of care treatment. We have long-term evaluations, and there's minimal general toxicity, and we didn't see any off targets. So we are now moving the technology to go to phase 1 clinical trial. More in general, what our technology about delivery, so the Aadi technology, so it's based on short amphipatic peptide, which forms stable complex with different type of payload and mainly nucleic acid, go through the cell via the non-endosomal pathway. We can deliver a large type of molecules, mainly nucleic acid, but covering from CRISPR complex up to DNA, mRNA, and small molecules. We show that we can get very potent Cas9 complex delivery and mRNA in vivo. We show that we can get very robust gene editing and genetic rescue in vivo, and there's no minimal toxicity and no cytokine activation. And the technology, the Aadi and the Daphtirex, is covered by a broad patent portfolio, which cover both the composition and the applications. Just before to conclude, to finish, I would like just to acknowledge people who have been involved in that work. In south of France, in Nîmes, on the platform Descartes, the people working on the design of the Daphtirex, people in Grenoble working on the animal evaluations, people at GenPep for the chemistry, and our funding. Thank you for your attention. I'm ready to answer any question.
Thank you, Gilles. While we wait for questions, can you explain why you do only 2 doses? Usually you see multiple doses in tumor studies is pretty common. So any comment on that?
Yeah, I mean, we can go to more than 2 doses. We did 2 doses because if we analyze after 2 doses, we didn't see any tumor reemergence. But I mean, the protocol would be maybe to use one, or instead of 2 doses, to go to 2 or 3 or 4 doses to be sure that we can knock down all the KRAS mutants, because we still have a background of KRAS mutants inside the tumor.
Okay. And a follow-up, in the clinic, do you anticipate that you would have limited dosing, just like in the animal study, or do you think that that you would do repeat dosing in the patients? Given that you're essentially deleting the function of the gene. So,
yeah, I mean, in theory, deleting the function of the gene is permanent. So it should be permanent for during the treatment. But I think in the clinic we will have to do multiple doses because KRAS mutation is occurring, and maybe on metastasis there may be more KRAS mutation occurring. So we may have to do multiple, I mean, repeat doses after the first treatment.
There's a question from Natalie, and the question is, was there modification as regard to the initial peptide for siRNA delivery to obtain the specific Cas9 delivery?
Yeah, I mean, we have some peptide working for the siRNA. For the specific, for the delivery of mRNA, Cas9, and guide RNA, we increase the amphipathy of the peptide. So that's one thing. And we have this targeting motif, which is not present on the siRNA peptide.
Okay. Okay, I think we should move on. So the next speaker that we have is Dr. Jasper Metzeler from the Department of Experimental Molecular Imaging in Aachen University Clinic. And he's going to speak on the clinical experience with liposomal glucocorticoid formulations for targeting inflammation. Jasper? You're on mute, Jasper. You need to unmute yourself.
Yes, thank you, Neil. Okay, thanks to the organizers for letting me, giving me the opportunity to talk about corticosteroid liposomes clinical experience. Just let you see what I could have looked like if I would have stood in front of you, and now you have to deal with this scruffy, just out of bed, just barely taking a shower face of mine. And that's what it is, right? It's we all feel lonely and we miss each other. So looking forward to meeting you in person next conference in Basel. Let's move to the contents of what I'm going to talk about. Corticosteroids, in the first place, in pendulated, long circulating liposomes, larger than we'd seen before in the earlier talk, and it's all about inflammation and passive targeting. So just the liposome by itself that goes to the site of inflammation. And this is what a schematic drawing looks like, talking about our strategy. If you have a free corticosteroid, you infuse that in the patient, it goes everywhere in the body, and very little actually goes to the site where it needs to be. Its corticosteroids have a PK that entails a very short half-life, big volume of distribution, so little target accumulation. And if you put it in a liposome, we tend to refer to that with the word nanocort. Liposome corticosteroid composed of PEG layer outside the lipid bilayer, 100 nanometers, so bigger than Stefan's, which was 35 nanometers. But we need that, it's maybe good to say here, we need this large particle size to just encapsulate enough corticosteroids. If we would have gone to smaller liposomes, we would have had a lipid to drug ratio of maybe around a factor 100. So we would have ended up with a product that would have to be sold in vials of maybe 200 milliliters or so, right? Big, huge infusions that we would have to give. And I'll come back to that infusion and the lipid dose later on in the lecture, so in the presentation, so that is an important thing to keep in mind. Now, if you infuse the liposome, obviously it stays within the blood circulation and it preferentially localizes at sites of inflammation. Now, we collected loads of preclinical results with that product, with liposomal corticosteroids. And I'm not going to show them here. I just show a few highlights, and I like this picture a lot because it shows where the liposomes actually go. We have seen pictures on nanomedicine localization before in this session. Now here you see where they go in sites of inflammation. You see a joint cavity on the left, a healthy joint cavity with a synovial lining. I hope that this pointer works. You see this synovial lining go here from one cartilage layer to the other, just a little bit folded. And now if you look on the right-hand side, you see an inflamed joint. You see lots of blue cells here accumulating around the synovial lining that basically goes here. Joint cavity is barely visible anymore. And you see that the blackening shows where the liposomes go, is sort of co-localized with the synovial lining. That's where most of the macrophages are. So indeed, macrophages play an important role in the uptake of the liposomes. And Cristianne already told in her lecture that in her case, around 33% in the tumor, where the 30% of the macrophages in the tumor that take up the nanomedicines. Now here it's probably around 90 to 100% even. So very important target cells. It's important because macrophages play a key role in inflammation, right? So it's actually good that we go there. They produce a lot of pro-inflammatory cytokines, and that's what we want to down-regulate with corticosteroids. Now here in this picture you see what it does in terms of efficacy. We saw in the animals macroscopically great benefits, much better than free corticosteroid. But here you see it at the level of histology. You see the joint cavity on the left-hand side again. Joint cavity, you see the blue cells again. You see the synovial lining here. And you see lots of blue cells. You see even blue cells. And you see a lot of exudate blue cells, inflammatory cells, that go into PMNs. A lot of them are PMNs also, go into this joint cavity. Now, if you treat the mice, in this case were mice with experimental arthritis, if you treat them with the liposomal corticosteroid, you see barely any infiltrate anymore, but also not any exudate anymore. You also see that the cartilage layer is left intact. So suggesting that doing this delays cartilage damage as well. It's very early in the disease here, so we've pictures collected after weeks in these animals and seen that indeed the cartilage layer remains protected. It's not affected by the inflammation. That's really important from a patient perspective. Obviously, you want to slow down the progression of the disease. Here you see inflammation treated with free corticosteroid. You see that that's much less efficacious. So we're talking rheumatoid here, arthritis, inflammation in the joints, joint inflammation, right? So just to highlight where that cell infiltrate is. Now, taking that to the patient level, RA patients, because it's liposomes, It's all known, and corticosteroids are known, so the step towards patient treatment is relatively straightforward. Not so much ethics issues, not so many toxicology issues to deal with. So we could do that first in a small study, 22 patients. I'll show you some results here. It's a study done in Nijmegen, the city of Nijmegen, the group of Piet van der Riel and Pilar Pereira. And we showed in that group of patients, you see a dose escalation basically here. You see the blue is a low dose, the purple is an intermediate dose, and the red is a high dose of liposomal corticosteroid. You see what it does at the level of the AS score, and that's a joint inflammation score in rheumatoid arthritis patients. Now you see that there is sort of a dose response visible, and one shot of liposomes shows efficacy over a couple of weeks' time. It's one shot. Now, normally you would have to treat these patients with daily oral doses of around 40 to 60 milligram of prednisone per day. And here you give one shot of 75 to 150 milligram. That has efficacy over a couple of weeks. So that's an interesting outcome. Oh, hold on. Now, pharmacokinetics, we've seen these pictures before also in this session. Half-life extension, she told you that free drug has a half-life of a few hours, low plasma concentration goes everywhere, so big, large volume of distribution. And you give the liposomes, it basically remains confined to the blood circulation. Has a half-life in our case of 3 days. It's a bit shorter than Stefan's 92 hours, remember? So we are at around 70 or so hours. A little bit more maybe. And you can measure the liposomes after single shot up to 42 days after treatment. There's a huge increase of area under the curve that we see. But it's important to make the remark here that all this area under the curve that we observe is all liposomally encapsulated corticosteroid. It's not efficacious. It's not available fraction. So it needs to go to the joint, and it's also shown here in patients where a patient had inflammation in the right elbow, right wrist, left ankle, left knee. You see with labeled liposomes, in this case technetium labeled liposomes, technetium 99M, showing in scintigraphy images that it really goes to the site of inflammation selectively. Accumulates there. And that's where the macrophages are. They eat up the liposomes, seen in the previous slides. They free the corticosteroids and make them available for action. That's the hypothesis. And does it only work in arthritis? No. We also did a study in bowel inflammation. Here you see a graph on the left, 17 bowel inflammation patients, ulcerative colitis patients, visible here at baseline screening, treated with the liposomal corticosteroid product and with a placebo. See strong inflammation, severe inflammation. These patients were severely sick, really, around the highest score possible in this disease. But if you treat the patients with the liposomal steroid product, you see a sort of black and white picture, and you see that a part of the patients, around 70%, shows a very good response, treatment response, and 30% of patients don't have any efficacy. So interesting. If we are only able to find out which patients are going to react, are going to respond, that would be, it would be wonderful, of course. So far, we haven't gotten any clue of what this black and white response could be a result of. Also here, seen with labeled liposomes, scintigraphy, that it goes to the inflamed lesion in the bowel. So targeting, passive targeting, EPR-based targeting, passive targeting, to macrophages. Okay, now we were able to take this to a higher level to expand the patient population in arthritis. We worked together with a big company, Sun Pharma in India. Together we did a big study in 150 rheumatoid arthritis patients. We had consultation with the EMA before, and they sort of, allowed us to make this one phase 2/3 study to help us progress this to product registration and build a dossier. Now, this is the setup of the study. We again did three treatment. We did 3 treatment study arms. One, but now the difference is 2 doses, whereas the previous study was only 1 dose. We did a study again in 150 milligram, the highest dose that we've done before, and 75 milligram. Again, to see whether there is a dose response and what is actually a good ratio, a good efficacy to safety ratio here, because we had too little evidence of safety of this product yet. Also, as a comparator, Depomedrol, that is a depot product, as the name suggests. It's an intramuscular product that slowly releases corticosteroid in the circulation. So the idea was that if our product, our liposome product, would only work through slow release, then it should show comparable results to this depot product, this Depomedrol product. Now, the dose is a little bit lower with the depot product as compared to the high-dose nanocort product, and that's because the depot product is made of methylprednisolone. That's a little bit more potent. So these doses are equipotent. The highest doses are equipotent. The study was called Esperance together, and so is the company, our company, with Sun Pharma in India. Now, here you see what happens in terms of EULAR responders. What is that, EULAR responders? Well, that's a defined patient population that has a minimal arthritis score reduction and some other key benefits that together define a patient as a EULAR responder. And you have good EULAR responders and moderate EULAR responders. And the good EULAR responders are, of course, the ones that we are interested in and that we set our end point in the study to. Now, you see here that our product is shown in blue and in orange, and the depot control product is shown in gray. And you see during the first couple of weeks that we have an increased number of responders. Quite a lot, actually, good responders, but also quite a few observed with the control product, more than we expected, basically. So that was interesting. Now, this efficacy advantage seen over the first couple of weeks of the study sort of diminished over time. Patients were followed for 12 weeks. So we had hoped, of course, to see a longer benefit, but still we were quite happy with this outcome. Here you see the DAS score with the inflammation score reduction over time, and it sort of confirms the picture in the previous slides. See the control product going down quite impressively, I have to say, in black here, quite impressively, from a severe score above 5 towards a sort of acceptable score between 3 and 4. And with the nanocort product, liposomal product, you see, and even with the lower dose, the lower dose is also more efficacious than the higher dose of control product. You see that it's significant also, and that you have quite a strong, fast reduction of inflammation in the earlier time points after treatment. And sort of this difference fades away slowly later on during the study. There's all sorts of other things that are important to tell here. I have put in a picture here that shows the cumulative EULAR response, good EULAR response. So the patient's population that builds up in every trial arm, that is a good EULAR responder. And you also see here that it goes slower with the control product and with the liposome product, it goes up quick. And you also see a dose response in terms of that the blue curve is better than the red curve. The higher dose is better than the intermediate dose. Okay, so say it again, Neil.
2 minutes.
Yes. Okay, so to round it all off, we have also looked at safety. It was important, right? You see here that comparing the safety of 3 products, there is not much of a difference seen. We're all having these kind of adverse events, mostly minor, hypertension, viral infection, mild weight gain maybe. We did not really see the typical corticosteroid-related side effects at the level of glucose, osteoporosis, and these kind of things. Addison's, these kind of things we didn't see. And that's interesting because they were typically seen if you treat patients with high oral doses. So that's good. That's good news. However, we were interested also in seeing what the infusion reactions, liposomes are known to cause infusion reactions. We spend a lot of effort in finding what are better protocols, infusion protocols. Now I come back to this issue of having a high lipid dose. We figured that the higher the lipid dose, the higher the chance of infusion reaction. So you need to sort of do something to the level of rate of infusion to not so quickly expose the body to the lipids that the liposomes entail. So this was an interesting thing to look at. And we still observed quite a few infusion reactions, even moderate and severe. The moderate and severe were not seen with the control products, as you see in this picture. And this may also be well due to the fact that we did not pre-treat the patients. So that is an important issue. We didn't want to do it, so we want to have a clear picture of it, what could happen. Now, for subsequent studies, we definitely need to pre-treat patients. Okay, so these are the phenomena that we observed with infusion reactions. And I'll round off with concluding, where are we now with this product, with this liposomal steroid product in inflammation, in arthritis, in bowel disease? Well, PK nicely confirmed, targeting confirmed, efficacy improvement confirmed, safety also confirmed. We need to do something about these infusion reactions. That's a little bit of homework that we still need to address and need to do before we go back to the EMA. We've seen, and I'm not showing that here because there's no time, but we have other indications with a high inflammatory indication, high medical need, in which you see strong efficacy. So that's encouraging as well. Now, in the rheumatoid space, there's a little bit of hesitance of whether we should continue, because, what is the actual niche that this product can take among other drug products in rheumatoid? What pricing can we get? And would insurances be willing to reimburse that? That's a major issue that we have not yet solved. And those questions basically stalled the further development in arthritis. So if you have comments there and input, please let me know. That is very, very interesting. And there is an online debate in the afternoon in the debate lounge where I would like to pose this question to you, and you can comment there. So if you have the availability and the interest in that, you please join me at 1 o'clock there. Okay, so thank you for your attention, and big thank you to all my colleagues and patients who have been involved in the studies. Thank you.
Thank you, Jasper, for that interesting talk. So let's see. Don't see any questions yet. Maybe I can ask one question. What is the composition of the comparator product, the Depomedrol? What does it have in it, or is it lipid-based, or can you say something about that?
Yes, it's a suspension. It's a suspension. So the steroids form a depot in the muscles and need time, the crystals need time to dissolve, and then are taken away by the blood circulation in the muscles slowly and build depot formulations in the blood of the patient. And that's interesting because the to take as a control, because I didn't show the PK data here with regard to free drug, you can imagine, of course, you anticipate high concentrations of drug in the target site, but at the same time, the drug leaks out of the liposomes over time and forms low concentrations of free corticosteroid there. So that could theoretically also contribute to efficacy. I just wanted to know whether this targeting advantage that we anticipate really shows at the level of efficacy. That's why we chose this control product.
There's a question from Dan Cromelin, and the question is do you have any clinical data on chronic administration, so more than 2 injections?
Interesting questions. No, not in
steroids as well.
Sorry, say it again, Neil.
And the follow-up question was: have you tried other corticosteroids as well?
Yes. Yeah. So this was basically all about prednisolone. We only administer this product twice, not more. We have another product in development that is a dexamethasone liposome product, and that's a product that we currently evaluate in cancer patients. Dexamethasone is given there in several indications. It is interesting whether we could increase efficacy or reduce safety issues there. And in one of those studies, we administered the product 10 times weekly. So that is way more frequent. Yeah. Okay. I don't have any efficacy results to show that, to see whether that in the long run does anything. Those studies are still too small to conclude, to draw conclusions from in terms of efficacy.
I have a question. In terms of your comparison with the Medrol product, is that data that you have that after 2 injections, it seems better at first, but then by week 12 or so, things equalize? So is that sufficient, that early drop in inflammation or improvement in inflammation compared to Medrol? Is that sufficiently beneficial to, or is that enough of a clinical benefit to further develop this product?
Yeah, very, very good question. Yeah, we had a discussion with the EMA also about it. And there were rheumatologists there as experts in the committees. And yeah, eventually we concluded that that could be the real key benefit of this product, that to help rheumatoid patients through the active phase of the disease, you can, for instance, start to give methotrexate. That's relatively safe and also quite efficacious. It doesn't have any steroid-related side effects, what most people are afraid of. So an aim is always to reduce corticosteroid adverse effects. If you can do it with methotrexate, then people prefer methotrexate. So it's actually a very good drug. However, methotrexate only starts to work after 8 weeks or so, 10 weeks. So it takes a lot of time to bridge those patients to that time of efficacy of methotrexate. Now, and that's really about this time, this bridging time, which you should do there for the patient. Now, most people and most rheumatologists stick with oral prednisone, but they really need to administer 40 to 60 mg per day to bridge the patient to efficacy of methotrexate. And then if you give so much, you give cumulatively more than 1, sometimes 2 grams of prednisone over that time, that really has detrimental side effects. So that's bad. If you can do this bridging with only 1 or 2 shots of liposomes with much lower cumulative dose, showing no typically corticosteroid-related side effects, then that's good. So it needs to bridge this one, and it needs to be quickly efficacious. We have patients that reported benefit within 2 days. One patient, remember, said she was she was working in the garden. She was lifting stones 2 days after her treatment. Well, of course, that was very, very much discouraged by the rheumatologist. But it shows that some cases can be very quickly efficacious. So that is really something to go for here.
Okay, thank you. We just have a minute or two left, and I'm looking at some of the questions that rolled in for some of the other speakers. So, Gilles Divita, are you still there?
Yes.
Okay, so there's a question from Marisa Papaluca Amati. So, did you manage to assess the duration of effect on primary cancer cells versus metastatic cells?
Okay. Yes, we did. We show that on primary cancer cells we have a strong effect, and when we look for metastasis, if we do a third treatment after that, we can see a reduction of metastasis. But it's really, as I said during my talk, it's really dependent on the KRAS mutations, and sometimes some other mutation can occur in the metastasis.
Okay, thank you, Gilles. There's a question for the small liposome. So Stefan, so how do you manage to prepare such small liposomes? You don't have to give away all your secrets, but you're welcome to respond. I think we have lost Stefan, so we don't have an answer to that question. Let me see what else. Okay, I think that's it for this session then. We'll thank all the speakers and thank the audience for participating, and have a wonderful rest of the plenum meeting.